In this video, I'll introduce the IGV Web App. The IGV Web App is a lightweight, online version of IGV that makes sharing data views easy. The IGV Web App is different from the IGV desktop application in the following ways. It runs in a web browser and doesn't need to be downloaded or installed. Sharing sessions and views is extra easy. And it has fewer features, but it supports the most popular use cases from the IGV desktop application, and more functionalities are continually being added. The IGV Web App can access online data via a URL or a file storage platform like Google Drive or Dropbox, as well as whatever local files you give it access to. Note that no IGV Web App user data is uploaded to an IGV server. The IGV Web App runs completely in the browser. There is no server behind it. Now that we understand the basics, let's open up the IGV Web App and load some data. You can select the reference genome in the Genome menu. You can either load a predefined reference genome from here, or load your own from here. Here we'll use the default Human Genome 19. Then you can load data using the Tracks menu. As you can see, data can be loaded from a local file, Dropbox, Google Drive, or a different URL. You can also load a predefined track from here. Depending on what reference genome you selected, there may be many predefined tracks or none. Here we'll use one of the Illumina Platinum genomes as an example. Navigating through data in the IGV Web App is very similar to navigation in the IGV desktop application. You can jump to a chromosome with this dropdown, enter a gene, region, or even a series of genes or regions in the search box to be brought there. Click on the cytoband to jump to a new region. Sweep across the genome ruler to zoom into a specific site. Click on the genomic coordinates to have that region fill the window. And zoom in further using the Zoom In button. You can also use the zoom slider to quickly zoom in or out of your view. You can focus in on a particular variant and right-click to sort by base. By right-clicking on the data, you can see options that will be applied to the data location where you clicked. Clicking on the gear to the right of each track will show you options that control that track. For example, you can color the reads by read strand and set track height, color, and name. Using the buttons on the upper right, you can hide or show the track labels. Enable the center guidelines, and enable the cursor guideline. Now, because we initiated this session by opening igv.org/app in the browser, refreshing the browser would return to the default page. So if you like what you see and you want to save it, you can either save your session in the form of a JSON file, bookmark your session, or just save an SVG of your current data view. And if you want to share this data view with someone, it's as easy as copying this URL and sending it to them to view in their own browser. Another video dives deeper into sharing data views, so for now, let's just make a bookmark of this view. Now if we reload this page, our view comes back. So if we want to close this window, we can always return to this session using our bookmark. If you need any help troubleshooting or you have more questions about the IGV Web App, click on the Help link to be brought to the IGV Web App documentation.